They become hematopoietic blood forming cells, stem cells. So HSC, hematopoietic stem cell. But the hematopoietic stem cell is more differentiated than a fertilized egg, from the beginning. But they are not well differentiated. They give rise to the three classes of blood cells. These are what? Leukocytes, erythrocytes, thrombocytes. And the other names are white blood cells, red blood cells, and platelets. Okay. And producing, it says the differentiated white and red blood cell, differentiated from the stem cell. That is what that means. Is a rare work of hematopoietic stem cell and their progenitor cells. It's calculated that, this is the number I was looking for. Great, it's here. So in the course of producing the mature circulating blood cell, the original hematopoietic stem cell will undergo between 17 and 19.5 divisions. And in the end, will give a net amplification of approximately 170,000 and 720,000 cells. So from one stem cell, you can get 170,000 neutrophils. You can get 720,000 neutrophils out of one stem cell. So we have few stem cells in our body, but we have a lot of white cells. We have a lot of red blood cells, simply because you take one stem cell and within a short of time, you produce thousands of one particular cell out of them. So again, the multipotential cell here changes into myeloid or lymphoid. You get the natural killer cell. You see the granules in the natural killer cell, and they are larger cells than the T lymphocytes. And then the small lymphocytes becomes T and B. B develop further to plasma cell, which secretes antibodies. You come to this side, the myeloprogenitor cell include the erythrocyte, mast cells, and then the myeloblast, out of which we have the basophil, neutrophil, eosinophil, and monocyte. Okay, and then macrophage. Okay, then the platelets are right down there. All right. Okay. So I was thinking somebody may ask if, for example, out of one stem cell, you can get 3 different cells out of them. All right. Is it a good question to ask? Okay, let me ask what you think.
Prof,
Yes.
initially you said that it depends on the growth factor that they are exposed to. That makes, that will decide the kind of cells you develop into.
Yeah.
So if you have the growth factors are targeted towards production of neutrophils, they will develop into neutrophils. If it is towards maybe a lymphocyte to become lymphocytes.
Okay. So yes. So when you look here, you see common, meaning that when it changes from this to that, this cell here can become any of this one. So it's a common progenitor cell. It can become any of this. But depending on what is exposed to, as is it Robert? Your name is saying
Richard.
Okay. As Richard is saying, this then converts to one of these. So this cell cannot be red cell at the same time as cell. So depending on what the common progenitor cell is exposed to, it can become a myeloblast. When it becomes a myeloblast, again, it has a chance to become any of these cells. Depending on what the myeloblast is exposed to, it will become eosinophil and not a neutrophil. And so eventually you get one type of cell out of them. But what we cannot predict is that if at the myeloblast stage, okay, as I said, the divisions and the increase in number, if you have 100 myeloblasts, all of them may not become a eosinophil. But the one that is converted to become eosinophil won't become a neutrophil. So if you look at it that way, if at some point the common myeloid progenitor cell converts to myeloblast and there are 100 of them, all of them will not be eosinophil, because between this and that, the markers are not for eosinophil, they are for this stage only. So the growth factor that converts myeloid progenitor to myeloblast is specific between these two and against these other ones, because at this point, this cell can be any of these four, but it can become only one. So when it's becoming a myeloblast and it's dividing and it's like 1,000 myeloblasts, at that stage, all 1,000 may not be eosinophil. Depending on the exposure, some can be neutrophil, some can be basophil, okay? So here we have the, again, now you see more stages. So if you look at this point, let me go back. Here, 1, 2, 3, and you are at eosinophil. That's not what happens exactly. So from a myeloblast, there are different stages that will lead to the functional cell. So eosinophil, there are also other stages that have been not shown here. So the cell doesn't just jump from myeloblast to eosinophil, but they go through different stages. Therefore, any myeloblast that changes to neutrophilic promyelocyte may not be able to become eosinophil. It is now committed to follow this line all the way to that end. The one that changes into eosinophilic promyelocyte, that kind of thing, will not go back to a basophil. Once they commit into the lineage, then they follow. But at the common stage, then they have the potential to become any of the other cells. So they are different levels. Don't forget, I said it divides between 17 and 19.5. And at any point, it differentiates. So at stage 16, the cells there are different from the cells at 17. At 18, the cells are different from 17. And then 19 is the final one. So maybe you will put division 16 here, 17, 18, and 19. And then you have your neutrophil. Okay. So the idea is for forming the mature blood cells that a precursor cell at a particular stage is that the receptor for those cytokines. So if the cell wants to convert to, say, a neutrophil and it needs interleukin-3, then that cell will produce interleukin-3 and receptor, and then link up with interleukin-3, which will stimulate the production towards a neutrophil, all right? And then it will switch off the production of receptors for the cells that it doesn't want to become. And then it will follow that pattern. So which differentiation pathway a precursor cell takes is thought to be a function of which cytokine it first encounters. And that's somewhat a matter of chance. But you can rule out the matter of chance when you look at the fact that the body also upregulates the cytokines that they want to be used to stimulate the production of a particular cell line. So therefore, the differentiation signals that a cell receives at particular times in its lifetime. The signals that the cell receive in its lifetime is crucial factor in determining whether it will become a white cell or a red cell, lymphocyte, or a macrophage. Okay. All right. And through series of careful studies of cultured cells. So earlier I mentioned that when I hit the laboratory, I may be able to produce a neutrophil out of them. And so to understand how these cells behave, these culturing systems were done in the laboratory, and cells were exposed to different growth factors, and they were able to differentiate and grow into the functional cell that we're seeing. So what we think happens in human beings can be replicated in vitro. Okay. So investigators have discovered many key growth factors and cytokines that induce progenitor cells to make different type of blood cells. Example, tissue, tissue growth factor alpha, tissue growth factor beta, and tissue necrosis factor alpha. They all do different things. Okay. And now you can have a lot of the growth factors. Yeah, you can see them. Even this is not an exhaustive list. Okay, so you can see the growth factors that if you want the stem cell to convert into common myeloid, expose them to interleukin 1 and then 3, 6, granulocyte monocyte colony stimulating factor and stem cell factor. So if you expose this cell to these factors exclusively, they'll convert this to that. When you expose this to GM-CSF, they'll convert the myeloid to a myeloblast. Expose the myeloblast to interleukin 3, 5, GM-CSF, and you will get a neutrophil. Okay, eosinophil. All right. So that is how the cell development goes. These proteins are inside our body, growth hormones are inside our body. Our body produce more depending on the cells that it has gotten. Earlier, I was mentioning the fact that the myeloblast can also become a neutrophil. So if you have 100 myeloblasts and 20 of them encounter these factors here, then the 20 will become a neutrophil. And then the rest will become eosinophil. So from the myeloblast, there is still the chance of they becoming the other cells, depending on what they are exposed to. So these factors interact with one another in complex ways to create a system that is very exquisite with genetic control and coordination of blood cell production. Through this exquisite blood genetic control, blood cells are developed through this system. And so, say the hematopoietic stem cell is a multipotent cell that can give rise to cells that are more and more committed to differentiate into a given cell type. So they differentiate into RBC. So, but the stem cell doesn't make hemoglobin itself. So for your body to get hemoglobin to carry oxygen, the stem cell must change into a red cell carrying hemoglobin. Okay, that it will at the same time remain stem cell. That is where they say they renew their base. Okay. So in developing embryos, blood formation occurs in aggregates of blood cells and this thing, I think if you have seen some of those questions, you may have seen it. The blood cell development begins in the yolk sac, or which are also called blood islands. That is where the cell development starts. And then as the fetus development progresses, the blood formation occurs in the spleen, liver, and lymph nodes. These are something you should take note of. So the development occurs in the spleen, liver, and lymph nodes. Okay. Then when the bone marrow develops, when the baby now develop bones and bone marrow, then the bone marrow will assume the task of forming most of the blood cells for the entire organism. Okay. But maturation and activation and some proliferation of lymphoid cells occurs in what is called the secondary lymphoid organs, such as the spleen, um, thymus, and lymph nodes. Okay. But when it comes to the immune system, we categorize the thymus as a primary lymphoid because the cells in immunology, when we say primary lymphoid organ, we are talking about where the lymphoid cells are developed. And then secondary lymphoid organ, we are talking about where the cells function. Right. Right. But in immunology, the cell development is bone marrow, and anywhere else is said to be a secondary cell development site. So in children, hematopoiesis occurs in the marrow of the long bones, such as the femur and tibia. Okay, and then in adults occurs mainly in the pelvis, cranium, vertebrae, ribs, and the sternum. Sternum is on the chest. In a healthy adult person, approximately 10 to the power 11 and 10 to the power 12 new blood cells are produced daily so that you can maintain a steady state. And these cells are shipped into circulation. So 10 to the power 11, 10 to the power 12 are produced on daily basis, mostly to replace dead and damaged cells. And that is in a normal person. All right. So zooming into stem cell maturation, we will see those blastocyst things very soon. Some pictures that will show you what stem cells develop into and what can be produced out of a stem cell. So say as the stem cell matures, it undergoes changes in gene expression that limit the cell types that it can become and moves it closer to a specific cell line. I've mentioned that as they develop, they move towards the specific cell line based on the exposure of the molecules or the growth assistant proteins that they receive. These changes can often be tracked by monitoring the presence of proteins on the surface of the cell. And each successive change moves the cell closer to the final cell type. When you change, when you were in university, anytime you move from one level to the other, you were getting closer to your graduating date, wasn't it? So the same way, any change, successive change in the cell that is developing will lead to that cell getting closer to the final functional cell type, okay? And because of that, it will limit the potential for them to become any other cell. You know, as I was saying, when you have learned science in secondary school, science in university, and then you have gone on to specialize, you are moving away from being able become any other professional. As I sit here, I cannot say that I can become a lawyer in the next 6 months. It's not possible. I didn't even study law in the university as an undergrad. And so the things I learned moved me to specialize in hematology and immunology. So like I said, as you progress, you are narrowing down towards a specific. Same with cell development. As it progresses, the cells are being shepherded towards a particular cell type. And once they pass a certain stage, they cannot come back to become that cell again. So here we see the fertilized egg. So that is an egg and sperm fertilized here. And then it divided into 2, 4, and 8. The 8th stage, 8-cell embryo. This is where any experiment can be done when you mash up this fetus or embryo and release the stem cells. And at this point is where you find the blastocyst. Blastocyst covering the cells inside. If you take these cells and culture them, you can develop neural cells, cardiac muscle, or blood cells out of this stem cell. But generally, it is out of this stem cell that the human being is produced anyway. And these 8-cell complex can yield into a human being, where the brain is developed, the head is developed, the hands are developed, amazingly. So also in terms of research, I can take stem cells and develop red cells out of them. These procedure can be done outside a human being. The only reason these things are not allowed now is that ethics is against experimenting with a fetus, and this is said to be on its way to become a fetus. So when you break the blastocyst and release this content, you are actually preventing a life from taking place. And so ethics is against it. That is why we can't develop these components. But a lot of research is still going through